This conference will now be recorded.
Hello, everybody. My name is Nathan Cohn with Texas Public Radio. I want to make sure everybody can hear me okay. So for those of you with your cameras on, our presenters, if you could just raise a finger and let me know. Great. Okay. Well, I see a lot of people on the call here today, and we're waiting for just a couple of seconds here to let everybody join in before Bonnie takes it over and the rest of our presentation starts. But I'll start by saying thanks for joining us today for this first ever virtual Think Science presentation on how vaccines work. I'm Nathan Cohn, Vice President of Cultural and Community Engagement for Texas Public Radio. And like all of our Think Science events, the goal with this one is to get you in and out in about an hour, having learned a little bit more about the topic at hand. And for those of you who've attended our Think Science sessions in the past, I'm very sorry we couldn't provide a box lunch for you today. Hopefully next time. I want to thank our sponsors for this Think Science event, and today's Think Science event is made possible in part by Baptist Health Foundation of San Antonio, Texas Biomedical Research Institute, and the San Antonio Vascular and Endovascular Clinic. We have a trio of researchers to talk to you today, and our moderator and host for the discussion will be Texas Public Radio's award-winning bioscience and medicine reporter, Bonnie Petrie. Bonnie's also the host of our weekly podcast on the coronavirus and its effects. We call it Petrie Dish. So be sure and look it up on our website at tpr.org and subscribe to it on Apple Podcasts or wherever you get your podcasts. Your favorite podcatcher, look for Petrie Dish. And of course, Petrie is spelled P-E-T-R-I-E. A little tech before I turn it over to Bonnie. The Q&A session today will be virtual as well. You should see a chat or a Q&A box in your GoToMeeting window. And when it comes time for questions, you can type them into that box and I'll field them to Bonnie and our panel. And this is our first time trying a large interactive event like this online using GoToMeeting. So apologies in advance if anything goes a little bit awry, and thanks for your patience and forgiveness. And now I'm going to have Bonnie take it over from me, and our presenters, you unmute yourself when you need to, okay? Bonnie, take it away.
Take it away. We all know why we're virtual today. We're in the middle of a pandemic, and that has limited our ability to interact with one another. And a lot of people think that this is going to be the way it is until there's a vaccine, which is why we're here. And the title of this Think Science is how vaccines work. We'll touch on that. And we will also talk about the work of our three presenters today, who I would like to introduce to you. First is Joanne Turner, who's the Vice President for Research at Texas Biomedical Research Institute and the Executive Director of the Vaccine Development Center of San Antonio. Also joining us, Dr. Karl Klose, who's the director of the South Texas Center for Emerging Infectious Diseases and a professor of microbiology at UTSA, and Dr. Evelien Bunnik, who's the assistant professor in the Department of Microbiology, Immunology, and Molecular Genetics at UT Health San Antonio. Thank you so much for joining us. So we're going to start off with Dr. Turner, and I'm going to put it on you, Dr. Turner, to answer the question, how vaccines work. How do they work?
That's a really big question. But let me put this in simple form for you. Really what a vaccine does is mimic or fake a real infection. So we're tricking our immune system into thinking it's seen the real infection. And the normal responses you'd have to that infection would obviously happen, but it would be in a safe way. And so it really is just tricking our immune system to think it's been infected, drive the right response to control that infection, so that when we see it again, that response is already primed and ready in place to go quicker and control the real infection faster.
So we had a detailed conversation the other day about the immune system. And I'd like you to share some of what you told me with our audience. What is going on with this coronavirus and our immune system?
Yeah, a lot, and a lot of things we still don't know. So we are right at the forefront of a pandemic with something new. And so there's lots of things we're still learning as we go. When the virus infects us, it first will enter cells. in our bodies, and those cells will really kind of set off the alarm bells and tell surrounding cells that there's something dangerous happening in our bodies. And that switches on what we call our innate immune system. These are frontline cells. They show up, they try and kill whatever is around them that's dangerous. For this instance, it would be the virus. And if they can't do that, then they actually call for backup. And then they bring in cells that we call cells of the adaptive immune system. And the adaptive cells come in and they actually have the ability to remember what they've seen. So they can try and clear the infection. And if successful, the next time we see it, they're ready and much quicker, and they know the exact response that's needed to get rid of that infection from our body, which means that we'll often, for a second time, be infected and not even know it, assuming that we've been able to generate that protective immunity. So that protective immunity is the same if we do it with a vaccine or with an actual infection.
So humans have seen coronaviruses. We've had coronaviruses for, I don't know how long, decades at least, but probably longer. They were discovered decades ago. What is the difference between the coronavirus that might give us a cold and SARS, MERS, and the COVID virus?
There's a lot of similarities with MERS and SARS, and that actually gave us an edge when we started thinking about how to study them, to study this SARS coronavirus too. We took what we'd learned from MERS and SARS. They have a lot of similarities in how they infect us, how the disease occurs, but this one is much more virulent and spreads more quickly, and that's what's different about it. The common cold, even though sounds very similar, is actually a very, very different form of infection, and not essentially the same kind of dangerous infection that we have with SARS coronavirus 2.
And why is that? Do you know?
I might have to defer to one of my colleagues to see if they'd like to answer that, because I'm not sure I could.
That's okay. We can put
I see Karl.
we can put a pin in that, and maybe Dr. Bunnik can take that one, or Dr. Klose, but we're putting a pin in it. So what I'm trying to get to is we have human coronaviruses, and these are zoonotic coronaviruses, right? What's the difference?
Well, so I think a lot of viruses that like to live in other animals are adapted to live in animals. They're very happy there. They've been cycling through these animal reservoirs for a long time. And then for reasons that we don't always understand, they do jump to humans. And then they actually can be not very well adapted to us. And so they'll cause quite a lot of chaos. Our immune system's not seen them before. They've not seen our immune system before. Whereas the common cold has learned to live with us for decades and decades, right? It's adapted to be in us. It knows just how bad to make us sick without really killing us. And we kind of live in a nice kind of, it's not very happy for us, but we've learned to live together. Whereas this virus is now, something we've seen for the first time, and neither are adapted to each other.
So Texas Biomed jumped right in January and February working on this. What's happening over there right now?
We are busy. And so before we can even think of a vaccine, we needed to find a model to test it in. So we can't just put a vaccine straight in humans because we don't know if it's safe. So we need animal models. Texas Biomed by nature has a lot of biocontainment facilities, so we're well suited to do this work because this virus has to be worked at in a heightened level of containment to keep people safe. And then we also have a primate center on our campus, and so we have non-human primates available to us. And so we initially started looking for what is the right model, and we have no idea what the right model was back at the beginning of this year. We've looked at 3 different species of non-human primates. We're currently looking at a couple of rodent species as well, and we're not the only ones doing this. People around the globe are really racing to find the best models so that we now know that we can test a vaccine in to make sure it's successful. So we've completed pilot studies. We know which species work better than others. And now we've started running vaccines or therapeutics through those animals. And so we often work through contracts to do work for other people that have spent a lot of time developing new vaccines. And that's where we're at right now. And then the next phase for us is doing our own basic research. So our scientists are now gearing up to write grants to NIH to ask for that government funding to do basic research. So we have a much better understanding of the infection so that we're well equipped to maybe design an even better vaccine in the future.
So talk to me about the Vaccine Development Center so we can lead right into Dr. Klose's presentation.
Yeah, it's a center that's a San Antonio-based center. It's for institute collaboration. So that covers UTSA, UT Health, Texas Biomed, and Southwest Research Institute. And we're much like the precision therapeutics that many people have heard of. We work in a similar way. We're collaborative. The Vaccine Center's primary goal is to share information about vaccines. We do that primarily through an annual conference. We really hope will occur this year as people can be mobile again by November. Also do support basic research studies. And we work with our community to manage, kind of interact with the community and educate people about vaccines. So this year, with COVID-19 coming about, SAPPT and us co-sponsored a project, and SAPPT sponsored projects on coronavirus in general, but we had one that was on vaccines, and so we felt like we wanted to support that project from the Vaccine Development Center. And Dr. Klose's project was the one that was.
Thank you for that, Dr. Klose. Welcome.
Thank you.
All right, take it from there.
So my laboratory, so I'm a researcher, and my laboratory has been studying an unusual bacterium. So I'm a bacteriologist. I am not a virologist. I wanted to just say that up front. So I understand infectious diseases. I teach microbiology, but I'm not a virologist per se. But my laboratory has been studying vaccines for a very long time now, and we have an advanced vaccine that is geared towards preventing the disease called tularemia. So it's an unusual disease. Most people have never heard of it, and most people don't even know anybody who's ever even had it before. So you might wonder, why are we developing a vaccine for something that nobody gets and nobody knows about, right? But the reality is that tularemia is on this watch list by the government as a potential bioweapon. And it has the same level of danger and government sponsorship as anthrax and plague, which everyone has heard of. So it's a very dangerous organism, and the level of threat is equal, I would say, to anthrax and plague. And so we've been developing a vaccine against this strange organism to protect people against the illicit use of that organism. And so because we have this advanced vaccine platform, I'm going to call it, and when the pandemic arrived, you know, around the world, we thought, well, why can't we use our platform to generate immunity against coronavirus? Because there's different types of vaccines, and a lot of vaccines that people are familiar with are what we would call subunit vaccines, where just a little piece of the organism is given to the individual to mount an immune response against it. So like diphtheria vaccine is made with a little piece of the diphtheria bacterium, and then that's what you generate your immune response to. But some vaccines, you really can't generate the correct type of immunity without providing the entire organism, okay? And so what that means is that our vaccine is actually composed of a living organism. It is not tularemia per se, it's a very closely related organism. And what happens is the bacterium that we use as a vaccine is basically crippled. It can't cause disease at all, but what it can do is it can induce the correct immune response once it gets inside of a vaccinated individual. But what that also means is that it's a living organism. And so living organisms, because of genetic engineering, you can force them to do things that they wouldn't naturally do, but we can basically design in the laboratory for this organism to now express additional pieces of additional diseases. So we've tried this technique before with our vaccine against the plague. So if we get the right piece of the plague and we put it into our vaccine, then our vaccine acts essentially like when you go to the doctor and you get a syringe, right? So our bacterium is essentially acting like that syringe, and instead now going into an individual and providing the right vaccination to vaccinate against additional organisms. So in the corona pandemic and this new SARS-CoV-2 virus, one thing that's pretty clear is that if you want to be immune against this disease, you are going to have to generate an immune response against what's called the spike protein, okay? And the spike protein is what you see on the surface, like when you see those pictures of the virus and there's those little like flower-looking things on the outside of the virus, those are the spikes. And the spikes allow the virus to get into cells. They are kind of like a key. You know, when you go to a lock and you put a key in and then you turn it and you get inside, these are the equivalent of a key that the virus contains on its surface, where it's looking for a cell to get inside of. And so it's pretty clear that, you know, maybe there's additional things associated with immunity, but minimally, we have to generate an immune response against that spike protein. Because what that will do is when the virus tries to get inside of a cell, antibodies will coat that spike and prevent it from getting inside of the cell. So what we're doing is we're genetically engineering our vaccine so that it will express the spike, not the whole virus, just the spike from the coronavirus, and then vaccinate, you know, animals and see if now they're protected against the coronavirus. And, you know, again, this is all theoretical, but there is a lot of basis for what we're doing in other systems and other vaccine approaches. And so we think that it will probably be successful. We just have to figure out how to express it in the correct way.
Thank you for that, Dr. Klose. Let's go straight to Dr.
I would like to just mention, because I forgot to say, we're doing this in collaboration. So I'm at UTSA. We're doing this in collaboration with the UT Health, San Antonio, as well as Texas Biomedical Research Institute and Southwest Research Institute. So all 4 institutions are involved in this effort to create a vaccine against coronavirus. I apologize for not saying that earlier.
That's great. Now that level of collaboration is really exciting, and I'm really pleased that it's all coming out of San Antonio. So Dr. Bunnik, he talked about immunology and he talked about genetics, and those are your areas of expertise, and you're working on antibodies research. Tell us about that.
Yeah, so we are very interested in studying the immune response that happens in people when they go through a natural infection. Because like Dr. Turner said, the response that you would want to elicit with a vaccine mimics the immune response that happens in a person during infection. And so we try to understand what that immune response looks like to then also better understand what kind of an immune response a vaccine should elicit. And one of the main questions that we're interested in is, we know that people who go through natural infection are probably protected for a certain period of time, but at this point it's very unclear how long that protection lasts. And also, if you design a vaccine that mimics that response, how long does that vaccine then give protection against infection? And at what point would you need to revaccinate people to try and to maintain that level of immunity? And so we work with blood samples from people who have recovered from COVID-19, and we basically look at 2 different things, because immunity is there are 2 layers of protection in your immune response. The first is antibodies. So antibodies are molecules that circulate through your body or through the blood, and that will bind the virus, the spike protein in particular that Dr. Klose already talked about, and prevent that virus from being able to attach to a cell and enter a cell to then start its replication cycle. So that's the first level of protection. And I guess you've heard a lot about the antibody tests that are now becoming available. So that's something that you can measure fairly easily, although those tests measure just antibodies in total, which not all antibodies are necessarily protective. But the second layer of defense that your immune system puts up are memory cells. And this is also something that Dr. Turner mentioned. So these are cells that have seen the virus and have responded to the virus and are sort of a pre-activated state. So the next time they see the same pathogen, they'll respond very quickly and generate a new immune response that is able to contain that infection without letting it get out of hand, as it would have happened the first time around when those cells weren't there yet. And so we're working on designing probes or reagents that we can use to track those cells in the blood of people who have who have been infected. And so we can look over the period of up to a year, over 2 years of time, see how many of these cells are present, and are those stable, are those cells maintained, or do they decline very quickly? And that will give us an idea of how long these people will be protected against reinfection. And that's essentially what we're working on. And those cells will also, these are the cells that ultimately will produce new antibodies. So we can also take the small part of the genetic material that encodes those antibodies and learn more about the type of antibody responses that these people produce in response to infection.
That makes me think of something I read today about we're working really fast to try to get a vaccine, and there are some thoughts that we won't be able to get a sterilizing vaccine, which is a vaccine, obviously, as you know, but our listeners won't know, that will make you completely immune to this virus, and then it might be one of those that just makes the disease less awful. What are your thoughts on that, Dr. Bunnik?
Yeah, that's a great question. And at this point, there's still so many things that we don't know about this virus that we have to leave every possibility open. I think yeah, I don't know what to think. That's
okay.
Where, I mean, I can make an educated guess, but that would just be about it. I mean, I think it should be possible to generate sterilizing immunity, because what we see now in people who have had COVID-19, that it seems that these people are protected against reinfection, and so I think we should be able to mimic that response and provide sterilizing immunity, at least for a certain period of time. But I think the big question is, how long will that protection last? And is that a year? Is it 5 years? And that's one of the things that we don't really understand very well about vaccines. Why do some vaccines give you protection for 20 years, and other vaccines you have to repeat? Like tetanus vaccine, for example, it's recommended that you get a booster shot every 5 to 10 years. Whereas for some of the vaccines that you get during your childhood, you essentially don't need a booster for the rest of your life. And there's still a lot of things that we don't understand about how vaccines work.
Well, there's a lot we don't understand about how the coronavirus works.
Exactly.
We're all flying blind in this pandemic in every aspect of our life, so don't feel bad about that. But I did see Dr. Turner's finger go up. So do you have something to add?
Yeah, I thought I would just add to that. I mean, we're in an absolute race right now to get an effective vaccine. And you hear on the news all the time that there's so many companies testing their vaccines, and we're trying to compress the schedule from development through testing through animals, through safety tests in humans, so that we can get an effective vaccine. And I think we will certainly meet the timeframes that scientists are talking about, which is about 12 to 18 months. And we'll most likely have a vaccine that's good. But typically what happens after that is there's a refinement stage. And that's why the basic research is occurring. That's why Evelien's work is so important. We need to understand what true protection is, so that we can now go on and develop an improved vaccine. And that will often happen against any infection. The first one is good enough to really prevent this pandemic, but we may have to come out again later with something that's much more effective, that's gone through a couple of years of research to understand exactly what's needed for long-term protection. And then those will come back later. And so the research won't stop. And as scientists, we always say it's going to take a year and a half. And I don't think people in the public want to hear that. We'd love to think that we could get it in a couple of months, because it's so critical. I think the thing to focus on instead is that there's an incredible number of scientists working on this, including myself. We've turned our normal research on other topics to this virus because we really want to find a vaccine. So it seems like a long time, but actually what we're doing is very, very compressed for how science normally works.
So is the idea of a warp speed vaccine in a couple of months realistic? You could take that.
I mean, I don't know what warp speed means, whether that means a week or a year or 10 years. I think it's a difficult word to use. It sets expectations very high. I think what we really need to be thinking about is how we can buffer and protect our population while our scientists really work very hard to find a good vaccine. And that may take a while. So the physical distancing, everything that we're doing is so important to kind of flatten that curve and give the scientists time to do it. So if warp speed means a year, maybe.
I know that all of everyone I've ever talked to in your field has been prepared for a pandemic for your whole careers. So this pandemic is really no surprise. I'd like to ask each one of you, if you want to answer, is this what you thought it would be like, Dr. Turner?
Yes, I think so. I think it could have been a lot worse. I think you'll find a lot of people would say this is our practice run to make sure we're ready for the next one. And so that's good because we've learned some really good lessons from this. And SARS and MERS before that were also our practice runs. I think this is what we expected. It typically will lead to a lot of disruption globally, even in the economy, healthcare. That's what we would have expected. What we couldn't predict was what form it would take. If we had unlimited resources, we might have been ahead of this because we could potentially have been screening for this virus long before it really became a problem. And that's, again, right back to where basic research is important. If we had the resources to look sooner, we may have actually had a decent vaccine before we really got to this point, because we would have been ahead of the curve.
Nick, is this what you thought it might be like? A pandemic?
To be honest, I don't know if I had given this possibility a lot of thought until it happened and hit us all right in the face. So my research is, before this pandemic, the main focus of my research is malaria, which is, it's a parasite, but it also kills a lot of people every year. And so I'm used to thinking about, pathogens that cause a lot of death and cause a massive burden to healthcare systems. But those are not, or malaria is not a disease that affects the US, right? So, and it's not something that we in the US live with. But of course, there are a lot of places in the world where people do live with a lot of infectious diseases all the time, and people get infected all the time. But personally, for me, I had never given it, I hadn't given it much thought how it would be if, when a pandemic would hit us. And so it's been, I think, for all of us, it's been kind of a roller coaster these past couple months, trying to adjust the way we live, trying to adjust the way we run our labs, but also just try to put a lot of effort into the research to try and understand as much as we can about this new virus. So it's, yeah, it's been, like I said, it's been a roller coaster ride. It's fascinating and horrifying at the same time.
Right, because you're not just researchers, you're people, you're human beings with lives outside of, your labs. So exactly. You've got to deal with all that too. Dr. Klose, your thoughts?
Yeah, this is, I mean, it's classic pandemic that scientists have been warning about for a long time. It has been anticipated that a respiratory viruses are the ones that usually pop up and transmit, jump into humans. And so the idea of a respiratory virus causing a pandemic has been talked about by scientists for a very long time. And so that aspect of it, I would say, isn't that surprising. However, I think the speed at which it basically circled the Earth was astounding. And as a scientist who works in the United States, I'm going to say that I was probably a little bit, I'm going to say naive, or maybe shunned the idea that it would take over, basically our entire society, because I'm in this kind of bubble where scientists, at least they act like they've got everything under control. And so I just kind of assumed that, the CDC and the World Health Organization would make sure that this thing didn't take over the United States and our society, and we'd have to shut everything down. I thought that we would have better control of it when it arrived in the United States. And so that was really kind of I'm going to say a sad wake-up call, that we really weren't prepared for something like this. And I do agree with Dr. Turner that this was a great dry run that tells us exactly where we need to focus our efforts in the future, so that when another pandemic arrives, and it will, that we're better prepared. My laboratory studies cholera, which is a pandemic that has been going on since the 1800s. So we're also very familiar with pandemic diseases, but pandemic diseases that affect primarily developing countries and are waterborne are actually much easier to control than airborne diseases. And so like this is sort of, I think, a wake-up call for everybody, that we are extremely vulnerable to these types of diseases showing up, and that hopefully in the future we will be better prepared. Specifically with testing, is there's the number one thing that screams out, is that we have to be better prepared with testing capabilities if we want to nip this thing in the bud.
All right, I think it's time to take people's questions. I don't see you, Nathan, and I really don't know where to look for the questions. So if you'd like to pop on and help me out here, that'd be great.
Yeah. Bonnie, there's a question here, and I would, for Dr. Bunnik, who says, what number of neutralizing antibodies are needed to elicit long-lasting response against the coronavirus, SARS-CoV-19?
That's a great question, and I hope to be able to answer that at some point. We don't know. It might be if you have, like, theoretically, when you so in the lab, when you culture the virus and you add it to cells, if you have one good neutralizing antibody, that can essentially inhibit the virus from entering the cell. So in theory, one antibody would be enough, as long as you have enough of that one particular type of antibody circling around. But what happens in a person when a person is infected is that you don't just get one antibody response, you get a lot of different antibodies, and some of them are neutralizing, some of them are not neutralizing. And that combined response is what drives virus inhibition and ultimately protection. And so at this point, we don't really know what a response looks like in people, and what's sort of the minimum number of neutralizing antibodies, or minimum number of footprints on the virus that need to be targeted by antibodies to get to protection.
Great question, great answer.
Questions are, again, if you have a question, there's a chat window up here, so you can type it in there, and Bonnie or I can relay it to the panelists. Another question, this is open to all, I suppose. It says, when a vaccine is developed, will it be tested specifically on older folks? As a senior, I worry a bit about older people reacting differently to a vaccine than younger folks.
Maybe I can take this first. I actually do research on aging, so I have a little bit of credentials. So most likely when the vaccines are first tested, they'll be tested in fairly healthy people initially for safety, and then they'll get rolled out to the general population. They will have to study older individuals as a specific group to make sure the vaccine's working in them. So historically, most vaccines don't work as well as we grow older. And so that's very likely to be the same thing here, that we may need to do a boost for our elderly population. It may be that they need to have a slightly different form of the vaccine to be more effective for them. And so we don't know the answers to that yet. We have been looking at young and old animals to see whether there are differences in even how they deal with the infection itself. And there are some subtle differences between how elderly monkeys, for example, might respond relative to younger ones. And so they're a very unique population to study. And I think they sometimes aren't thought of when we're in this race to make a vaccine, but we will need to pay attention to how it's actually protecting across different age groups, including in children, and look at those groups specifically, and maybe make some adjustments to the vaccines to work in those very specific groups in the future.
Okay, we have a question from Heather. Can the presenters explain how the body normally holds on to immunity to a virus after it's been attacked? Do our cells keep a portion of the DNA, RNA in our DNA, or does the memory cell mentioned hold on to it? And how is a memory cell different from other cells or something else? So many questions in there. Who would like to take it?
Yeah, I can comment on that. So it's indeed about the memory cells that we have talked about earlier. And so the cells that make antibodies are a specialized cell type we call B cells. And these B cells have receptors that recognize pathogens. And essentially every single B cell that is produced by your body is different, has a slightly different receptor. And so all of those B cells together create a really large repertoire of recognition, being able to essentially recognize every different pathogen, even pathogens that are novel, are newly introduced into the human population. There will be B cells in your body that are able to respond to those pathogens. And so when a B cell gets activated, so you have a large pool of just B cells circulating through your body, essentially looking for things that it recognizes, looking for things that are not human, that are not supposed to be there. And so when your B cells recognize something that's foreign, and just that recognition by itself actually is not important. There need to be some other signals from other cells. But when your B cell so your entire immune system recognizes something that's foreign, that B cell will get activated, and basically has two choices. It can either become a cell that produces antibodies, and that's how you end up with antibodies circulating in the blood, or it can become a memory cell that still has that same receptor on its surface, but it will be in sort of a hyper alert state. So it will still continue to circulate, but the next time that it recognizes that same antigen, it knows, hey, I've seen this before. Last time it was bad. This time it's still bad. I'm going to just pump out tons and tons of antibody, and that response will happen really fast, much faster than the first time around. And the memory B cell, the second time it will get activated, it needs less additional responses from other parts of your immune system to generate that heightened immune response the second time around. I hope that made sense.
Okay, yes, definitely. So next question is, how would our response to this pandemic be different if the mortality was 25% instead of the low percentage that it appears to be? Anyone?
It's a scary and good question.
Yeah, well, it's a scary question for sure. Dr. Taylor?
Karl, do you want to take this one?
I can talk about it. It's pure conjecture because, you know, we don't really know. But what I would suspect is that it would be much more stringent. So in other words, like this pandemic, because it targeted, I'm going to say mostly older population was the real mortality that was seen, has been seen, is older people, that there are different segments of the population that are, I'm going to say, more complacent. And I think that with a 25% mortality rate, there would have to be a very strong national response to a pandemic like that. In other words, there would have to be a national stay-at-home directive that's the law, and people have to stay home because otherwise lots of people are going to die. But again, pure conjecture. I also think that the 25% mortality rate, you know, I'm just going to say that every pathogen, it's kind of like a game that they play. So if they're more lethal, so like the Ebola virus is a good example. The Ebola virus is highly lethal, but it's not as contagious, if you'll notice, as the coronavirus. Coronavirus spreads very easily among people, but there's lots of people who don't even know that they've been infected. And so the pathogen is actually very effective at going from person to person because it's not killing off most of its hosts, right? So you can imagine, think about it from the pathogen's point of view. If you're a pathogen, all you want to do is replicate yourself and move on so that your kids and your grandkids and your great grandkids can also live, right? So in order to do that, you have to not kill your host. So the most successful transmission occurs when the pathogen doesn't cause as much disease in the host, but is spread effectively, right? So like the cold, for example, is spread extremely effectively through essentially every population of the world, but it doesn't cause any kind of lethality. And then on the opposite extreme, we've got Ebola, which has a very high lethality, but it's easy essentially to kind of wipe out those pandemics after they start because of the transmission route, right? It's actually direct contact with persons who have Ebola. So the pandemics are actually easier to contain because it's more difficult for the organisms to spread from individual to individual. So, I mean, if you think about it from the pathogen's point of view, this coronavirus is about optimal, right? It's not causing as much lethality as you might anticipate, but it's spreading extremely quickly. So in other words, it's replicating itself in all of the hosts that it interacts with.
It's extremely efficient in that way.
Yeah.
Do any of you know how long it'll be before we have some sort of accurate idea of what the mortality rate is of this virus?
Yeah, I can speak. I mean, the other panelists can talk about this too. But testing has been seriously lagging. But now, what's important to know is how many people were infected to begin with, because there's so many people who have no symptoms. They don't go and get tested for active infection. But what Dr. Bunnik was talking about is antibody responses to the virus, and those are maintained after the virus is long gone. And so these are these tests that they're talking about now for antibodies, looking to see who in the past was infected with the virus, right? And once you have that picture, then you know these are the number of people who were actually infected, and here's the subpopulation who actually got sick, and here's even a smaller subpopulation who died. So you can see the bigger picture of how this virus was being transmitted among people who didn't even know that they had it. So antibody tests are just as important as the tests that they have for direct virus infection at the time, which is what people are going to the drive-thrus and looking for active COVID infection. We're talking about antibody infections, antibody tests now. They've been doing that in a number of countries that are sort of past the peak of infection. Like, so Spain just released a large study last week where they were looking for, in different areas of the country, how many people were actually infected. And the numbers were way higher than the people obviously who had symptoms and came down with the disease. But it's on the order, I want to say in Spain, it was something like, in certain areas it was up to 10%, and in other areas it was below 1%. So they see this sort of variability among the population, and probably is dependent upon how dense the cities are, how much people interacted with each other before there were stay-at-home orders, that kind of thing.
A quick follow-up: how accurate are those quick antibody tests that are now being promoted out there?
I don't want to get into any, like, comparisons or whatever, because there's a lot of them. I'm sure you've noticed this. There's a lot of antibody tests out there. Everyone rushed to produce their own antibody tests. And my understanding is that only certain ones of them have actually really been vetted by the FDA. And so I'm not 100% sure like which ones you can believe, but I do know that, again, this is an area where the federal government needs to step in and say, okay, this is the antibody test. We're going to use it because we trust it, because right now there's a lot of different antibody tests on the market, as you all are familiar with.
There are dozens, and the FDA's only approved like 7 or something.
I know, right?
Yeah. So next question is one of the subjects of this week's Petri Dish podcast. So I'm excited to hear what everybody says. When a body is infected, does the immune system release a lot of different weapons, antibodies, in hopes that one will work, or is it smart enough to release just the right one? We talked about this, Joanne, earlier this week. Would you like to start?
Yeah, and maybe I can start, but certainly we have immunologists on the call with me. I thought I'd start by saying, actually, our bodies are really smart, and we know when we've been infected, and we often know roughly what we've been infected with. So most of the cells in our body have proteins on the surface that are really sensors, and they can detect if it's a virus or a bacteria or a parasite or a fungal pathogen. And so our cells are able to do that. And so there's never just a really general response. It's an educated response, even though it might be quite broad. So the cell may say, I know I've been infected with a virus. And it will start sending out all the right molecules that need to drive an immune response to viruses in general, or a subset of viruses. What it doesn't know is the exact virus, and it doesn't have a response specifically to that one virus. So the first wave of response is generalized, but not entirely naive, right? They know it's a virus. And then what happens after that is that the other immune cells come in and educate themselves to the exact virus that it is, the exact proteins that are important for the virus to enter our cells or cause damage in our bodies. And that's when you get that specific antibody response. So the first encounter with anything that's infectious, our body makes a really educated guess in what the response is and tries to make the right response, and then they refine it afterwards. And so that's when you get the antibodies coming up that are very protective. If you've seen it before, then obviously, yes, you immediately make the good response.
Dr. Bunnik, the immunologist, we haven't seen this before. So what is happening sometimes? We hear a lot about cytokines and cytokine storms.
Yeah, so sometimes when your body is trying to fight the virus, it kind of goes into hyperdrive and it does too much, and it ends up harming itself. So you can make too many cytokines, activate too many cells, and instead of just killing cells that are infected with the virus, you end up killing healthy cells and healthy tissue, and end up with a lot more damage than what was the original intent. And so this virus seems to be really good at driving that hyperactive immune response in some people, not in everyone. And as a result, your own immune system is basically harming the lungs or other organs in the process, which is obviously not a good thing. But again, there are still a lot of things that we don't understand about how exactly the virus does this and what we can do to stop it. But there seem to be certain cytokines that are particularly important against this or in this process. And if you can specifically inhibit those cytokines, you could essentially prevent the cytokine storm from getting out of hand and resulting in a lot of tissue damage.
Yeah, so a lot of the odd symptoms that we've seen in this virus over the last couple of months, some researchers attribute to these cytokine storms, from blood clots to acute respiratory distress syndrome. Does that make sense to you, Dr. Bunnik?
Yeah, it does. And well, to some extent, the blood clotting that's a really interesting phenomenon that I think puzzles many researchers and many physicians as well. They've seen people throw clots in places and in quantities that is completely abnormal. And young people, for example, that normally don't really have blood clots, have lots and lots of blood clots and get strokes and all those kinds of really weird things. But yeah, to some extent, that can all be related to what the virus is doing to the body and how the immune response, or how the immune system is activated by the virus, and like I said, generates a lot of damaging responses.
The next question sort of piggybacks on this, with the question being, why is there such disparity in severity among otherwise healthy individuals, with a lot of them being asymptomatic?
I think to some extent, at least what I've read about this, to some extent, it can be explained, and to some extent it can be explained by how much virus you are exposed to. So if you are exposed to a relatively small amount of virus, your immune system is probably able to contain that infection, and it can, so you can have a very local infection in your upper respiratory tracts, in your nose, and you have symptoms that are more similar to a common cold. If you are exposed to larger quantities of virus, it's more likely that the virus will be able to get down into your lungs, and that is what then gives more severe disease symptoms. But that, I don't think, explains everything. And again, there are still a lot of unknowns that we will have to figure out over the course of the next years, probably, as to exactly how the virus drives disease and why certain people seem to be completely asymptomatic and other people die from this infection. It's really, there's a large disparity. It's very interesting.
Nathan,
Yeah,
would you like, oh, go ahead, Dr.
can I just add something to that? It was a great point. What I would say is that, you know, maybe some people don't appreciate, you know, a brand new pathogen like this. There's so much that we don't know. And a lot of the information that comes about happens after the fact. So there's a huge population. One thing you have to appreciate is that the human population is extremely heterogeneous, right? In the laboratory, we can learn something about a mouse, which is, you know, some, you know, specially bred mouse and what happens in controlled environment. But out in the open, in human populations, there's so much heterogeneity. But the virus is actually telling you something as it's passing through individuals and really hitting somebody really hard, and then other people don't even know that they were infected. So those types of studies happen after the fact, right? So scientists will look at the people who were severely affected versus the ones that weren't affected, and then figure out, well, what is different about those people, right? What is different about the ones who are more susceptible? And a lot of times it may be something that's not right currently known, or it could be something that maybe the individuals didn't know about themselves. For example, some genetic predisposition. So you can imagine, like in the general population, people discover a gene that's associated with enhanced risk for cancer, right? They didn't know about that before. But now that they know that there's a genetic marker that makes them predisposed, then all of a sudden things become clearer. And I think that something very similar will happen with this virus, that the individuals that were hit really hard by the virus, at some point in the future, it will become maybe more clear why those individuals were more susceptible. We just don't have the data and the information yet, but we will eventually come up with the reason why some of these individuals are affected.
So early on, there was some talk about blood type impacting susceptibility, which would make one think, made me think, I don't know if it makes everybody think about genes and genetics and susceptibility. What work is being done on that?
I mean, I'm going to defer to my colleagues, but I would just make the point that there's so much work going on in this virus you can't even imagine. So what you have to appreciate, and I'm going to make a little plug for scientists here. So what you have to appreciate is that scientists are problem solvers by nature. That's why they go into science, because they like to solve problems. And all of a sudden this pandemic shows up and all scientists are hopping on board because they want to solve the problem, right? There's a huge, you know, global crisis, and yeah, they're members of the Earth as well, but they want to solve it for everybody else because that's what they're specialized in doing. So there's lots of scientists all over the world right now trying to figure out these questions that are being asked, which are all excellent questions. And we will discover the answers to at least some of them in the very near future. And the other thing I would also make a little plug for scientists is that nobody's essentially being paid to do this. These are scientists who dropped everything that they were doing so that they could solve the problem for everybody else, because that's what they're trained to do, right? So I know that, you know, frontline nurses and doctors, understandably so, get lots and lots of credit for being on the front lines trying to combat the infection, but there are scientists and labs all over the world that are trying to solve this problem as well, and they're going to come up with the solutions for the future.
Agreed.
Shameless plug for scientists.
That's why I love scientists, because I started covering this story in January, and you all are working together so well and so beautifully. It's really inspiring. It really is. I really think it is. Would anybody else like Dr. Bunnik, genes, genetics, genetic susceptibility related to virus?
I mean, we know for a lot of other viruses that there are genetic determinants that that influence how a person responds to infection. As an example, for HIV, we know that some people are almost resistant to becoming infected with HIV, and that's a genetic determinant. And we also know that some people have a much longer period of time when they are asymptomatic, so it takes a lot longer for these people to develop AIDS, whereas other people progress to AIDS much faster. And I'm sure that we're going to see similar genetic differences that can explain why some people, in the case of SARS-CoV-2, have much milder infections versus people that have more severe disease. But at this point, I know there have been a couple studies, and honestly, I can't remember exactly what these studies reported. There have been a few studies that had some preliminary results that address this. But like Karl said, I think we're going to hear a lot more about this in the coming year.
I predict they'll be writing about this for the next 100 years, at least.
Maybe so, yes.
You know, I'm reading a Spanish flu book right now, and that was more than 100 years ago. That was written this century. So, Nathan, would you like to read the next question?
Yeah, we're coming up on time with about 3 or 4 minutes left here, but enough time to answer this last question here, which I think is a good way to kind of close it out, because Heather asks something about what we as the public can do. And her question says, what can we as the general public do to facilitate antibody testing of as many people as possible to determine the true rate of spread / mortality, but also to get as much info as possible to the scientists so that you have what you need to work on finding a solution and getting these vaccines? Who do we talk to? Is it lawmakers, nonprofit research funders? What can the public do? Who should they be getting in touch with?
I think all of the above. So we need the public to make some noise on our behalf. It's great for us to know how many people test positive, but we still also have people who are getting really sick from this infection. And so that's going to be our focus, to make sure we have a vaccine. But certainly that information is important. But I think making noise about making sure funding gets to scientists so we can do our job, that we have the support we need and have the information that we need. And also making sure that when you seek out information, that you go to the right sites online and you get really good quality, accurate information and not some of the misinformation that's out there. So help us educate your friends and colleagues with the right information, help educate our politicians that really need to put money behind us so that we can do the work, and get that testing out there so that it's accessible in every single physician's office so we can get that data.
Would anybody else like to add anything on that final question?
Or just nodding in agreement?
Yes. All right, well, is that it, Nathan?
I think that's we're coming to the close here. And so Bonnie Petrie, thank you so much for moderating today. And Dr. Joanne Turner from Texas Biomed, Dr. Karl Klose from UTSA, Dr. Evelien Bunnik from UT Health. Thank you very much for being a part of this panel today.
Thank you for having me. It was a pleasure.
Yeah, it was a pleasure to be on the show.
So much
We want
fun.
to thank also our sponsors for Think Science, Baptist Health Foundation of San Antonio, Texas Biomedical Research Institute, and the San Antonio Vascular and Endovascular Clinic. This session is being recorded right now, as you heard at the beginning, and so once we have the file and we're able to post it online, then we'll put it up on the tpr.org Think Science page, so that if you want to share this session with any of your colleagues, your friends, your family, you can do so. So thanks everybody for taking part in the first ever virtual Think Science panel, and I hope you have a good afternoon. Don't forget to listen to Science Friday on Texas Public Radio coming up in just about a minute.
Thank you.
Thank you.